Welcome dear viewers. I am Dr. Amit Bhattacharya from Department of Zoology, Ramjas College, University of Delhi. Today I will be speaking on a title which is Karyotyping from Chromosomal Aberrations and Abnormalities. So this is a very important topic which should understand for discovering all the diseases and prognosis and subsequent method of treatment regimes for these particular diseases. Now, before starting this one, there is a branch of science which is called as the cytogenetics, which is a branch of biology which focuses primarily on the study of the chromosomes and its inheritance. So, it is a branch which deals with the medical genetics. Now, in this particular branch, we study technique which is called as the karyotyping technique. So, if we see the image over here, this is a karyotype which has been shown, in which all the chromosomes are structures. They are then stained with the particular stains, and then they are segregated to form in a particular pairing. By looking at these particular things, we can tell about the abnormalities in this chromosome number or the structure. So, the chromosome number in many of the cases may increase or decrease also, and in many of the cases there may be some kind of aberrations or chromosomal deletions or inversion in these chromosomes. So, all these techniques and these particular things can be used up or studied through this karyotype method. Now, as I told you, the cytogenetics is a branch of science which deals with this focusing and study of these chromosomes and their inheritance. Now, before moving into what exactly this karyotyping is and how we check the chromosomal abnormalities, we should understand what exactly the chromosomes are. So, chromosomes, as we know, are the microscopic structures which contains DNA within them, and they reside within the nucleus of the cell. Now, during the cell divisions, these structures or the chromosomes becomes condensed and are visible to the through the microscope when we stain with the particular staining techniques. Now, these particular staining techniques are used to access the number and the structure of the person's chromosome, and a part of this forms the diagnostic testing also. Now, these particular diagnostic technique or the testing helps the scientist or the doctor to analyze what kind of disease the person is suffering from. Is there any genetic disease, or is there any chromosomal changes which is occurring within this particular person or the patient. Now, chromosomes, as we know, are the thread-like structures which are located inside the nucleus of the animal or the plant cells. Now, each of the chromosome is made up of protein onto which the deoxyribonucleic acid is wrapped on. Now, the chromosome term came from a Greek word which means color, which is chroma, and the body which is called as the soma. So, scientists gave their name to the chromosomes because they are the cell structures or the bodies. They are strongly stained by some characteristic dyes which are used in the day-to-day researches. Now, in the plants and the animals, there is a designated number of the chromosomes which are there. For example, in the humans, we have got 23 pairs of chromosome, of which 22 pairs of the chromosomes are being numbered, and these are many a times referred as the autosomes. One pair of the sex chromosome is there which can be XX or XY depending upon the male or the female type. So, in total there are about 46 chromosomes which are there. Each pair contains two chromosomes, one coming from each of the parents, which means that the inborn child, it inherits half of their chromosomes from their mother and the other half from the father. So, both of them contribute for this particular thing. So, if we see the picture or the diagram over here, this particular diagram tells us about how the unique structure of the chromosome which wraps on with the help of the DNA, and there are pool of proteins which are there which are called as the histone proteins. Now, the histone proteins are octamer which is formed of various proteins onto which this DNA is wrapped twice in two turns, and these histone proteins are further then condensed into characteristic structure along with the DNA to form the condensed structure which are called as the chromosome. Now, these chromosomes are found within the nucleus. So, we can see over here how the hierarchy of this particular characterization is shown over here. Now, the DNA molecule is there, so it is unwound so that the transcription or the replication process can go about. So it is wrapped on the histone, so the unwinding of the DNA has to take place during this particular processes. Now, the chromosomal numbers vary among the living organisms. Most of the bacteria, as we know, have got 1 or 2 circular chromosomes or the circular DNAs with there. And in the case of the human, most of the animals and the plants, they have got a linear chromosomes. In fact, there are a number of sets of chromosomes which are there. For example, the fruit flies has got 4 pairs of chromosomes, while the rice plant has got 12 and a dog has got 39 pairs of chromosomes. In the human, there is 23 pairs of the chromosomes, while the first 22 pairs are called as autosomes and the last pair is called as the sex chromosome. Now, if we see the diagram picture over here, it is showing that how the mother, which is having 2 X chromosomes, it gives to the offspring, while the father is having 1 X and one Y chromosomes. Now, biologically, female people inherit 1 X chromosome from the father and 1 X chromosome from the mother, while biological male person, they inherit the X chromosome generally from the mother only. So we can see over here how the crossing over is taking place and what the child progeny can be, either male or the female type. Now over here, the chromosomes, as I told you, are of 2 types. One is autosome, the other one is the sex chromosome. So autosomes are basically 22 numbered pairs of chromosomes which are there, which carries almost most of the part of the genes of the cell, and they are present within the nucleus. So we can see over here all the chromosomes from 1 to 22 are shown over here, which are called as the autosomes. Now the largest autosome is the chromosome 1, which approximately carries about 2800 genes, while the smallest autosome is the chromosome 22, which contains approximately 750 genes. So these particular chromosomes may vary in their structure as well as in the length also. Now over here, if we see the diagram over here, the karyotype or the ideogram which is shown over here, we can see the pair or the autosomes which are shown over here. So they are being arranged in a fashion of their number 1, 2, 3, 4 like this one till 22, and then this X chromosome is there, which can be either X chromosome and Y chromosome or 2 X chromosomes which are shown over here in the case of the female. So the male has got 1 X and Y chromosome, while the female has got 2 X chromosomes which are there. Now the X chromosomes are basically, there are 23 pairs of the chromosomes as we just discussed in the human cells, of which the last chromosome pair is the sex chromosome. So the biological female individuals have got 2 X chromosomes, while the biologically male have got 1 X and 1 Y chromosome. However, there are some exceptions to this rule also. Now, the X chromosomes are about 3 times larger than the Y chromosomes, and it contains about 900 genes, while the Y chromosomes has about 55 genes present within them. Now, the Y chromosomes in the humans and other mammals have 1 pair of sex chromosomes in each of the cell. The females have got 2 X chromosomes, while the male has got 1 X and 1 Y chromosome, as we discussed. Now the Y chromosome is very small. It is likely to have about 50 to 60 genes present within them, and it represents about 2% of the cell's total DNA over there. Because only males have got Y chromosomes, the gene for these chromosomes tend to be involved in the male sex determination and developmental process. So they are very important for the male sex determination in the developmental process in the males. Now, depending upon the centromere, which is the point of attachment of the 2 chromatids within the chromosomes, they have been named or nomenclatured by that way. Now, the centromere was first described by a German biologist. His name was Walter Fleming in the 1880s, and it was initially called as the primary constriction of the chromosome. Without the centromeres, the cells cannot divide, or the chromosomes cannot segregate and move towards the opposite pole. So the overall process is dependent upon the role of the centromere for accurate segregation of the chromosomes. So it is very important part of the chromosomes. Now, on the basis of the location of the centromeres, the chromosomes have been classified into 4 major types. For example, metacentric, submetacentric, acrocentric and telocentric chromosomes. Now, the position of the centromere thus provide a very useful information about the dividing chromosomes and what the type of that particular chromosome will be. Now, the karyotype groups have been divided or standardized based on this particular thing, and they have been mapped according to the presence of the centromere at that particular location. Now, if we see the image over here, this particular figure or the image, it shows the metacentric chromosomes as well as the submetacentric chromosomes, the acrocentric chromosomes and the telocentric chromosomes. The metacentric chromosomes, if we see over here, have got two of the arms which are equal in size, while the submetacentric chromosomes, which is having one of the arm which is shorter than the other arm. Then there comes the acrocentric chromosomes, in which one of the arm is more shorter and one of the arm is larger, while the telocentric chromosomes, it lacks one of the other arm and there is presence of only one arm. So the centromere is at the telomeric end or the end position of that particular chromosome. Now, these particular classification have been done, and according to them, the chromosomes have been divided and they are arranged in this particular thing. Now, the acrocentric chromosome, as I told you, is having an arm which is extremely small and the other arm is extremely long. While the telocentric chromosomes, one arm is missing completely. So we can see over here, the short arm is called as the P arm, while the long arm is called as the Q arm over here. Now, this is a karyotype of the normal female karyotype, which is having 46 chromosome, the 2X which is there. So we can see all the autosomes which are present over here and the 2X chromosomes which are present over there. Now, the normal male karyotype, which is shown over here, so the 46 chromosomes are shown over here, while the X and Y chromosome, which are the 6 chromosomes which are shown over here. So all the autosomes are being shown over here. Now, the cytogenetic testing or the conventional karyotyping is a very important technique for understanding what are the abnormalities which are present within these chromosomes. Now, the cytogenetic testing, it helps the scientists to examine the chromosomes and determine whether there is any kind of chromosomal abnormalities which is there, such as the aneuploidy or the structural abnormalities. Aneuploidy means there is an increase of the chromosomal number or there is a missing chromosome number. For example, one or more chromosomes, extra chromosomes are present. Like for example, 46 is the normal pair, but in the person there are 47 chromosomes. While in many cases there is a missing chromosome, so the chromosome number decreases, like the 45, which is a XO syndrome which is there. Now, there are various kinds of aneuploidies which are there. So, for example, the Down syndrome, which is a trisomy of 21st chromosome, means there are three 21st chromosomes which are present instead of two 21st chromosome. Edwards syndrome, which is a trisomy of 18. Turner syndrome, which is a monosomy of X, so one of the X chromosome is missing over there. And apart from that, there are a number of structural abnormalities which is there. For example, duplication, deletion, translocation, inversion and the insertions which are there. So these are the various forms of the structural abnormalities which are there. Now, how exactly these cytogenetic testings are helping to diagnose the various diseases? Like for example, they play a very important role in the diagnosis and the classification of the leukemias. They help to estimate the prognosis, they help to understand the treatment regimes which are there, and subsequently it helps how the patient is responding to that particular treatment over there by carrying out the genotyping or the cytogenetic testing of that particular individual. So the cytogenetic testing is, as said, is an important diagnostic tool both in the fetal and the genetic medicine, oncology and hematology for understanding all these disease conditions. Now, if we see over here in this particular image, it shows how exactly the karyotyping is done. So initially about 5 ml of the blood is taken on, then the blood cultures are being done. So there is an addition of the phytohemagglutinin which is added, so it helps in the differentiation of that particular thing. So the cultures are done at 37 degrees Celsius for number of days so that the cell growth takes place. Subsequently, there is an addition of a colchicine which takes place. So colchicine is a chemical which inhibits the spindle fibers so that all the cells get arrested in the metaphase plates only. So the cells will be arrested in the metaphase because the metaphase chromosomes are the chromosomes which are best looking and they are the best represented structure which can be easily watched under the microscope. Subsequently, the cells are fixed and then they are treated with various solutions so that the cells breaks out and then they are put onto the slide where the chromosomes are arranged randomly. Now the image of that particular chromosomes are taken place and subsequently that image is with the help of the various softwares they are arranged based on the chromosome number. So we can see over here the karyotype is being formed over here. In this particular karyotype we can see all the autosomes and the sex chromosome has been arranged, and then under the microscope or the visual description of this particular thing, it helps the scientist or the doctor to understand what kind of disease condition the person is suffering from. Now as we see over here in this table, there are various banding patterns which are there. So these banding patterns or the chromosomal banding techniques, they help the scientist to understand or identify the specific segments within the individual chromosomes, what kind of aberration or chromosomal changes this is happening. So there are various banding patterns which are there, like the Q banding, quinacrine, then the G banding which takes place with the help of the strain Gimsa. So there are various banding patterns through which these banding pattern, there is a series of consistent landmarks and the length of the chromosomes are being strained, which are the characteristic feature of that particular chromosomes and which helps the person to understand this particular chromosome structures. Now if we see over here in this particular image, the various banding patterns or the chromosomal bandings are being shown over here, like the Gimsa staining technique banding is shown, the Q banding, R banding and C banding. So depending upon these banding structures, the various chromosomal structures are analyzed and the cytogenetic testing is being done of that particular patient or the person who wants his karyotyping to be done. Now the chromosomal abnormalities have been classified into two major broad terms. One is called as the numerical, the other one is called as the structural thing. So in the numerical abnormalities, there is either a one chromosome or a number of chromosomes are missing, or there is a presence of one or more additional chromosomes instead. So that is called as the numerical chromosomal abnormalities, where the numerical number of the chromosome increases. While in the case of the chromosomal structural abnormalities, there is some part of the chromosomes which has been changed over a period of time. So it can be insertion, it can be deletion, it can be inversion, it can be translocation. So based on what kind of chromosomal abnormalities or the structural abnormality has taken place, they have been classified accordingly. Now how exactly and why exactly these chromosomal abnormalities happen? So one of the most important phenomena why this happens is called as the non-disjunction of the chromosomes. As we know, there are two important mechanisms of cell division. One is called as the mitosis, the other one is called as the meiosis process. So in the mitosis, the two cells that are duplicates of the original cell. So one cell with the 46 chromosome, it divides and become two cells with 46 number. So the chromosome number stable and equal in both the cells. So this is the mitosis process. So it takes place throughout the body except the reproductive organs, while in the meiosis, in this particular thing, the cell numbers halves. So the cell number or the chromosome number becomes 23 instead of the 46 chromosomes, and it generally occurs in the reproductive organs, resulting in the formation of the eggs and the sperms. So these are very important phenomena for the reproductive organ divisions also. Now the chromosomal abnormalities are basically classified into deletion, duplication, translocation, inversions like this one. So deletion means, as the term is telling you, there is a part of the chromosome which has been deleted or it is missing over there. Duplication means a part of the chromosome is present in a duplicate copy numbers. So there is a extra genetic material which is present over there. The translocation is where there is a portion of the chromosome which has transferred from one chromosome to the other chromosome over there. Now there are various other structural abnormality which includes inversion. A portion of the chromosome has broken off and turned upside down and reattached over there. So there is a genetic material which has been inverted in that particular cases, and subsequently there are rings which are formed, which have been broken off from a chromosome, and it forms a circle or ring. So this happens with or without the loss of the genetic material. Now, one of the syndromes which takes place through these chromosomal or the structural variation is called as the Karyotyping syndrome. So this is a very rare genetic disorder in which a part of the 5p chromosome arm is deleted over there, or it is missing over there. So generally the symptoms varies depending upon the location of the related genetic material, but one of the few of the common symptoms is the child, it cries like a cat. So there is characteristic facial features, slow growth which is there, and there is microcephaly which is also occurring in these particular things, which is a condition which indicates that the head circumference is smaller than what is expected in an infant age or a sex. Now, these, if we see the picture over there, the translocation is shown over there. So now the translocation have been classified into two major terms. One is called as the reciprocal translocation, and the other one is called as the Robertsonian translocation. Now the reciprocal translocation, the segments of the two chromosomes have been exchanged. So we can see over here the two chromosome pair, they have exchanged their segments over there. So this is called as the reciprocal translocation. While in the Robertsonian translocation, an entire chromosome has attached from one centromere, detached from, and it is attached over there. So instead of two pairs of the chromosomes, there is three chromosomes which are there. One of the chromosome gets missing over there. Now the karyotypic analysis, it helps the scientists to first count the number of the chromosomes which are present in the particular cell, and this is generally done with the help of the metaphase spreads which we just discussed. So this metaphase cells are being used because we treat them with the colchicine, which helps the scientists to look into these particular chromosomes and look for a binding pattern for each individual chromosomes in the selected cells. So these particular things are called as the karyogram, and these is the cytogenetic analysis which is there. So with this note, I come to the end of the first part of the lecture. In the second part of the lecture, I will be discussing about the various syndromes and various diseases which occurs due to these chromosomal aberrations and chromosomal change in the chromosomal structures. So thank you very much for viewing this part of the lecture. Thank you.